So hello. Today what we're gonna do is I will first explain you about the second part of the core activity because you have to do the first part, the second part, and the final part. So I didn't explain you yet about the second part, so that's what I will start with. Then I asked you in the WhatsApp group, if you didn't do it yet, you can still do it. uh, I asked you to install the application which is called iCell because we will review today the cell organelles and we will use this application to do it. And then we will do,
Teacher,
yes, dear.
sorry, I just arrived. I was having internet issues.
Okay, they told me. Don't worry, Annabelle. Uh, I don't put your absence. So I was saying that we will review the cell organelles with this application, which is called iCell. And then we will start today with what you can see on my background is the explanation of the endosymbiotic theory, what it's all about. And it's important that we understand how a cell is made of and that we know which organelles it has and the parts it has, because this helps us to understand better the endosymbiotic theory. But first things first, and that's the explanation of the core activity. So I'll share my screen with you. So the second entry, it's about, you're going to investigate about human impact on biodiversity. Remember that in the first part, you were looking for definitions about ecosystems, about biodiversity. So basically, you have the terminology, what this means. And now we go a little bit more in depth to investigate what is the human impact in biodiversity. And more specifically, you will look at agricultural activity, because this is an activity that is very important because we need to eat, of course, but it has a profound impact on our biodiversity. Uh, Possibly more than industry, because agriculture takes way more space than industry in general. So what you have to do is you're going to divide yourselves in teams. And let me be very clear about this, guys. You did a great first entry, most of you, but it's important that everybody has a team. I know that some people said we don't like team activities, but this is a team activity. We have to learn how to do that, so you have to do it in teams. It can be the same team, you can have a different team. For me, it's the same, but you have to be three. Okay, and you have to mention all the names here, the matriculas there, and each team member has to hand the activity in. Let me show you that quickly. It is written in here. Can you see my canvas right now?
Yes.
Okay,
Yeah.
perfect. So if you go in modules, so don't forget to do that, because it's such a pity. There is a lot of students that lost points just because they didn't hand in a certain thing. So core activity, human impact on biodiversity, delivery to, look, delivered by each team member. Okay, don't forget that everybody has to hand it in, and you click on it and then you get this Word document. So let me go back to the Word document. It's again very simple, guys. It's really not big science. The whole clue behind this is that you learn how to do research. You're going to fill in the following chart: country, geographic location, cultural elements, hydric resources. I will tell you in a moment what your topic will be. You can choose. And then you have to answer this question. So I would like you to go point by point and make it very clear and very nice, and that you don't forget any of the points, okay? So also in your work, you're gonna write 4A this, 4B that, 4C this, so that you address everything, okay? It's very simple questions you have to investigate. I'm not going over all the questions. You can read them. If you have questions, you can ask me. This last point is very important. Don't forget the references, because I've seen in many cases that students don't put the APA format yet. It's a pity you lose points if you just don't do the references in the right way. You have to do that in the right way during your whole prepa, during your whole professional studies. And also, I don't know if some of you noticed, but you already get points for writing your name. So please write your name. If you forget to write your name, you get already 5 points less. That's a pity. Okay, so that's what you have to do. So how do you know what your topic will be? That's another document. Normally it's assigned, but I let you choose. You possibly have already seen it in the WhatsApp group. You can choose whatever you want, but I want you to write me an email. For instance, Isabella, Vera Sofia, and Maria Fernanda, write me an email. Look, the three of us is a team, Isabella, Vera, and Maria, with this matriculus, and we want to do about wine in China, for instance. Okay, and then you get it. Okay, the only thing is that the persons that select first, they get first the topic. So if Vera, Sofia, and no, Vera, Sofia is the same person. If Isabella, Vera, and Maria Fernanda say, we want to do wine in China, and then Luna, Ana Pau, and Gustavo say, we also do wine in China, I just check the time when the email arrived. The one who got it first gets it, okay? And then if Gustavo, Luna, and Ana Pau said, yeah, but we also want to do wine, well, take wine in France then, okay? But not the same thing in China, okay? So you can choose whatever you want. It's interesting to see it's about avocados, coffee, wine, and asparagus, okay? The four of them, maybe asparagus a little bit less, but especially the three first one have serious ecological impact on certain regions in the world. You investigate what happens with coffee in the Arabian Peninsula or in Chiapas in Mexico. There is issues there. So you investigate this and you focus on your area, okay? I think it's always interesting not to take things for Mexico, but if you really want to do Mexico, you're free to choose whatever you want, okay? But it's all there's in everything there is one thing in Mexico, but I think it's nice to learn about other countries, but it's up to you, okay? Any questions? You have it in the WhatsApp group, so I'm not going to share it again. In the WhatsApp group, you can see this for topics. You just select what you would like to do with your group, and that's and you let me know by email, and then I will send you back okay. And if I say okay, you can do it, okay? And if I say like, sorry, there is another group who already had it, please select something else, then select something else, okay? Is that clear? Any questions about this? No, no questions? Okay, perfect. So I can close this. It's very simple, guys. It's really the content you make. It's really about how much effort you put into it, how much pictures you put, how much content, investigation, different sources. It's very interesting. But if you do, the more effort you do, the more interesting it is, in fact. So I think it's a nice activity to do this. It's very multi because it's about the world, it's about different places. And you don't only learn about avocados, coffee, or wine. You learn about different places in the world, and you will see what the issues are there. Okay, so the next thing is I asked you to install. I asked it in the WhatsApp group some days ago. If you didn't do it, don't worry. You can install it in 2 minutes. It's not a very heavy application. Install iCell here. So I installed it on the computer. It's not working. All right, it was working before. Okay, install iCell on your phone, and then let's play with it. Okay, we will use it to review the cell organelles. So everybody has it? Yeah?
Yes.
Okay, let's dig in then. What you can see here is animal, bacteria, and plant cell. Okay, you need the three of them. I do recommend you
Yes.
to keep this application the whole semester on your phone. And if you really like it, you can keep it forever, but keep it the whole semester because it helps you learn about the cell organelles in a more fun way, let's say. So let's first go to animal cell. If you click on it, you can see an animal cell. You can turn it around. You can look what's inside it. It's 3 dimensions, so that's why I like it a lot, because it gives you a better idea of how a cell looks like, you know, than just seeing it on a picture or a drawing, you know. So that's why it's a nice app. Please take in mind it's very, very simplified. You know, it's really very basic. This cell is more complex than this, but this is what we have to know. Here in the left bottom, you see text, and you can click basic, intermediate, and advanced. Okay, I would recommend you to put it on advanced because it gives you the most information. But at this moment, what we have to know is just intermediate or basic. We will go into more details as the course goes on. But for this moment, we just have to know what is the basic function of the things of each organelle. So I'm going to keep it on intermediate. Okay? And you can do the same thing. So I would like to have your participation, Isabella Trussell. Uh, can you tell me what is this structure all about here, this dark structure, if I click on it?
It's the nucleolus or the nucleus?
The nucleolus.
The nucleolus, it makes the ribosomes. The nucleolus is part of the nucleus that makes the parts needed to build a ribosome.
Okay, perfect. That's it. In the nucleus, it's in fact the ribosomes are produced. Ribosomes is a type of protein combination RNA. It has different components, and they're produced here. They travel out of the nucleolus. They go through the nucleus, as you can see with my mouse, and then they go here to the edge of the nucleus, and they travel through the nuclear pore to the endoplasmatic reticulum. And when I zoom out, you can see here on the endoplasmatic reticulum, you can see those black dots. These black dots, and those are the ribosomes, okay? So let's go to the next part. Javier, or Xavier. Xavier is in French, Javier in Spanish. What is this all about? The nucleolus, and then the nucleolus is inside another structure, and this we call the nucleus. What can you tell me about the nucleus? If you don't know it, open your app and look in your phone what is written, okay? So try to use this app.
The ones that have proteins.
No, the nucleus has no proteins. Well, it has proteins, which we call histones, but those proteins the nucleus has is only to package.
I was in. Sorry, I was in another part.
Okay, make sure you select the correct one.
These ones have the DNA.
Yeah, exactly. The nucleus is the place where the DNA is stored, okay? About a human cell has about 1 meter of DNA in it, okay? So it has to be packed very, very carefully because we have billions of cells, and in each one of them, we have a piece of DNA. And think about that. A cell is so small, you cannot even see it. It's smaller than the point of the needle. And inside that, we have more than a meter of one molecule, which is called DNA. So it has to be packed. And even this is packed in a very small place, because if you look, the nucleus is only a very small part of the cell, and all the DNA is in there. And the DNA is in fact, is the genetic information of the whole organism, you know? So the more complex the organism, the longer the DNA, the more complex the DNA. Okay, let's go to the next one. So humans have very long DNA. This yellow one, Adrian, can you tell me something about this yellow one that you see here? I'm turning it around. Remember, it's part, you can clearly see it here. Remember, we said it's part of the endomembrane system because it has all these different kind of layers because it's made of membrane. What is this yellow thing, Adrian? Can you tell me?
Endoplasmatic reticulum.
Okay, and what is it doing, this endoplasmatic reticulum? What is its function?
It says, can I read?
Yeah, of course.
The endoplasmatic reticulum aids in the transport of proteins made by Ribosomes.
Okay, that's correct. Like you can see here, these black dots are ribosomes. Those ribosomes, they assemble proteins, and the protein is produced inside the endoplasmatic reticulum. And then the endoplasmatic reticulum, it's in fact a transport mechanism, but at the same time it folds, it connects, it does things with the proteins, you know, so that they can fold and that they can assemble correctly, because you know a protein is a chain of amino acids, and it still needs time, and it needs a little bit of distance to do its folding and its assembling in order to be a functional protein. Okay, the next structure is for Anasophia. What is this green thing here? Looks
That
very
is a lysosome, and it's a lysosome, and it's in charge of basically cleaning and taking out of the cell everything that's worn out, and just basically cleaning everything that's not needed in the cell,
okay.
and just
You get 100, Anna Sophia, perfect. It's inside. It's indeed the waste recycling plant. Amelia, you're next to. I mean, on the screen at least, you're next to Anna Sophia. What is this blue thing here? Depending on the
That's
cell.
the mitochondria. It is very popularly known with the sentence, mitochondria is a powerhouse of the cell.
Okay, perfect, Amelia. I'm glad you knew that. Mitochondria is indeed a powerhouse of the cell. Okay. It produces the energy. We will see later in detail what it does. We have another structure here. Perhaps Gretel, what is this here? This thing next to the endoplasmatic reticulum that looks with different colors a little bit. In this one, what is it?
Golgi body.
Yeah, what does it do?
The Golgi body or Golgi apparatus aids the modification, packing, and distribution of proteins that are made in the cell.
Yeah, exactly. So receives the proteins that are produced inside endoplasmatic reticulum, but they're still not 100% done. So in the Golgi body, what happens is they sometimes, certain proteins, they need still some sugars on top of it, or they need to be cut or whatever. This happens in the Golgi apparatus or the Golgi body, and as well, the Golgi body sends it into vesicles away to places where this protein is needed. This protein can be needed. So here, vesicles are released, and there's one thing you have to understand. Certain proteins are needed inside the cell itself, but other proteins, they travel outside the cell. It depends on the protein, okay? So the cell needs proteins for itself, but it also produces proteins for going outside the cell for doing other things, you know? Think about antibodies that are released by white blood cells in the plasma and in the serum. So those antibodies, they're not inside the cell anymore. They're proteins that are floating freely in the intercellular matrix in the blood, basically. Okay, let's go to the plant cell. We are not going to see each part of the plant cell because you know that they are exactly the same as the animal cell. But what we can do is quickly revise the differences. So, Giulietta, what was one big difference between plant cells and animal cells that we have seen? So this is a plant cell. How is it different of an animal cell? Except for the color.
I don't remember.
No? Who can help? Itzel, maybe you can help?
Chloroplasts?
Yeah, very good. Chloroplast. What you see here is a chloroplast, okay? Something particular that only plant cells have. In a moment, in the endosymbiotic theory, we will see where how they were formed. And the chloroplast, what is the function? What is it doing actually?
The photosynthesis.
photosynthesis.
Yeah, very good. It's doing the photosynthesis, and photosynthesis, like the word says, is transforming sunlight into matter. So it takes CO2 out of the atmosphere and assembles it into glucose with the help of sunlight. Okay. What else is the difference here is the cell wall. So in this cell, you cannot see the cell wall. if I say put cell wall, you don't see anything anymore because it's like this biomolecule cellulose that is covering the whole cell. So that's why we hide it usually. That's another difference. Okay. Luna, I know you just said something, but anyway, Luna, what is the last difference? We have the cell wall, we have the chloroplast, and there was one more difference. I mean, there are more differences, but the main differences, let's say.
I don't know, the vacuoles?
Yeah, very good. You do know it's the vacuole indeed. If you click on here, you don't see it very good because it's transparent, but here you see the vacuole. If you put it on the side, you can clearly see it. So it's like this this bag next to the cell. What was it doing, Isabella and Tolsma? You remember what the vacuole is for? Somebody else knows? Soju?
It is a membrane bound.
Stores water.
Yeah, not only water, also nutrients, waste products. It's a storage unit, okay? So it can store whatever the cell needs at a certain moment, okay? And then the last thing that we're quickly going to see is the bacteria. I think it's important to look at this because if you look at it, you see how simple it is. Look at this bacteria. It has very little things. If you compare it with the eukaryotic cell, like plant and animal cells, they are eukaryotic cells. This bacteria is a prokaryotic cell. And what you see is just like a structure, a cell membrane. Inside, you see this violet structure, which is the DNA, and you see some yellow dots, which are basically just the ribosomes that are floating around. And for the rest, it has not much things inside. It has another structure here, which we call plasmid. And plasmid is also just a piece of DNA. Bacteria, they have plasmids, and it's just a piece of DNA that is separated from the rest of the DNA. So they can inject this DNA in other bacteria, so they have a little bit of genetic variety, because the problem of bacteria is that each bacteria is a clone of another one. So so if you have one remedy like antibiotics to kill a bacteria, it will kill all of them unless the bacteria creates resistance. And the bacteria can create resistance thanks to the plasmids. That a certain bacteria has resistance, it puts the plasmid into another bacteria, and then a gene that one bacteria might not have, it's transferred to another one. So that's basically a mechanism how bacteria evolve. It's a little bit more complex than that, but that's just what you have to know. Another thing that you see here on this bacteria is that it has this tail like a sperm cell, you know, spermatozoid. We call that flagella. Okay, so certain bacteria, they move with a tail, which we call flagella. And this bacteria, it looks like oval shaped, it's long. But please keep in mind that bacteria can be round, they can have a comma form, they can be rod shaped, they can be rectangular, they come in all different kinds of sizes and shapes. But what they all have in common is that bacteria are really, really simple. And keep that in mind, because I'm going to explain you now a little bit how cells were formed and what is the endosymbiotic theory all about. And it's something really important because last lesson we were talking about, I mean, not the last one, but last week we were talking about origin of life. And the endosymbiotic theory is basically an addendum of the origin of life. Yeah, so let me I'm gonna charge the content here, put it in big. Okay, and we're gonna see it in 7 little steps. Okay, you just have to know the basic, guys. There's a lot of extra information here. If you click on the plus, you don't have to know that, okay? It's just for your information. The only thing I ask you to know is that when somebody talks about the endosymbiotic theory, that you can explain a little bit what you think this is, okay? So in the origin of life, we have seen that there are many theories of life. And in these origins of life, we also have seen an experiment which we call the Miller-Uller experiment. In this experiment, they have proved that if you take the right conditions and you put the right atoms in water and you take lightning and volcanic activity and heat and sun rays and whatever, that those atoms will automatically form biomolecules. Why? Because of the consequences of the laws of physics and chemistry, you know. The formation of this is the consequence of the laws of nature. Okay, so biomolecules had to form because energy is absorbed and atoms fuse. Okay, so this caused that biomolecules were formed. Biomolecules itself, because of the charges they have, because of the interactions they have, and the forces they have between each other. If you have phospholipids, one phospholipid will be attracted to another phospholipid, and they will automatically form a membrane. When this membrane becomes round, you get like a circular membrane, and you already have a cell. If there is other molecules that are by accident captured in this phospholipid membrane, for instance, like more complex molecules like proteins or RNA or DNA, then life comes into existence and basically you have a cell formed, okay? So our story of the explanation of the endosymbiotic theory starts when we assume that we already have bacteria on planet Earth. And we know that we already have bacteria on planet Earth for millions and millions and millions of years, even billions of years, okay? Bacteria, we know that as a fact that they were the first kind of organisms on planet Earth. Bacteria, single cell organisms, okay? One of the first ones were what you can see here, cyanobacteria, okay? And it's a very special kind of bacteria because it's a kind of bacteria that we still have today. It's a bacteria that was able to capture sunlight and convert it into matter, basically, okay? So what this bacteria did was using the CO2 in the air of the primitive Earth and used it to form matter, to form itself and other things, and released oxygen into the atmosphere. You all know when you did the mass extinctions activity, when you looked into the timeline, how were the levels of CO2 in the beginning of planet Earth? If you go millions and millions of years ago, in the beginning of the Earth, like 3 billion years ago, how was the atmosphere of planet Earth? Did it have a lot of carbon dioxide or little?
A lot,
A
because
lot.
of all the volcanic
A
eruptions.
lot.
Okay. Very good. You paid really well attention. Indeed, because of the formation of the Earth, there was a lot of carbon dioxide in the atmosphere. And this carbon dioxide, over millions of years, has been depleted from the atmosphere thanks to those cyanobacteria, okay? What they did is they captured the CO2, they used it to their benefit, and they released oxygen in the atmosphere. So the oxygen and the atmosphere we have today, we have it not because it was formed out of nothing. We have it because the first life on planet Earth made the atmosphere how it is today. If those bacteria wouldn't have been there, those bacteria that live in the sea, that live in water, and that do photosynthesis, then our atmosphere would be still toxic for us. Then intelligent or multicellular life would not have been possible, okay? So that's in fact the first step of the endosymbiotic theory. We made our own atmosphere. The atmosphere on planet Earth is made by life, okay? Keep that in mind. It were the bacteria that made the conditions on planet Earth as they are today, not volcanic eruptions or anything. Of course, they had a big part in it, but it were the bacteria that really changed the atmosphere. When those bacteria were releasing all this oxygen into the atmosphere, we got to a percentage that we have today, which is about 21% of the atmosphere is oxygen. We have a lot of nitrogen, and there's very little of carbon dioxide left. You know that we also have an ozone layer. Yeah, this ozone layer it wasn't always there. The ozone layer was formed. Okay, how was it formed? It was formed because the atmosphere was built up by those bacteria, and when this oxygen comes in contact with solar radiation and UV radiation, ozone can be formed. Okay, who knows the chemical formula of ozone?
O3.
O3, very good. O3 is something that we don't have a lot here on our height, let's say on the surface of planet Earth. We don't have a lot of ozone, and that's a good thing because ozone is very, very unstable and it's very reactive. If we would have a lot of ozone here on the surface of the Earth, then we would have severe respiratory problems. In fact, ozone is formed on the surface when you have a lot of pollution and a lot of sunlight. For instance, smog. There is ozone in smog. It's very irritating for us. Another place where we have ozone formed is, for instance, if you go to the photocopy shop, Copyca. If you go there and you have like 20 photocopy machines, they have very strong light, and ozone is produced with photocopy machines. So it's not very healthy to be a lot of time with a photocopy machine because they produce some ozone because of the light and the photocopies that they do. It's very unstable. It's very easily destroyed. Again, this ozone, it falls into pieces and it forms oxygen again, but it also forms ozone between space and the atmosphere, and we're very happy that that we have ozone formation there, because if we didn't, then a lot of this dangerous radiation from the sun would reach the surface, and that would be really bad news for us. Okay, so the ozone on the top layer of the atmosphere is protecting us. Guys, the ozone layer is really, really, really thin. Do you have any idea how thin it is? I don't know the exact number, but it's not more than a meter thick. It's really, really thin. It's something of nothing, and it has the capacity of absorbing all those dangerous radiation, okay? And of course, we had this big environmental problem in the 80s when we had a hole in the ozone layer in the South Pole. It formed for a particular reason in the South Pole. It's a whole other subject. I was very fascinated by it because I was, why is the ozone layer above Antarctica when they never use those chemicals that make the hole in the ozone layer? Well, it has to do something with the cold, of course, and with the vortex of the Earth. You can do some research about it, but it's very interesting how it is formed. In fact, the good news about the ozone layer that it is an environmental problem that we managed to solve more or less. Okay, so all the chemicals that cause the hole in the ozone layer, they are banned now, and we see the last 20-30 years we have seen a big improvement in the ozone layer. And we even think, the scientists even think that in the next decennia to come, that the ozone layer might be completely back to normal. So let's go to this endosymbiotic theory, because I see there's only little time left. So we had this bacteria, they made our atmosphere, and they made a condition that was more ideal for bacteria, okay? So if there is more oxygen in the atmosphere, you could have other types of bacteria, like aerobic bacteria, which means that a bacteria that use oxygen to their advantage. And this gives the rise to the first traits of early eukaryotic cells, okay? Early eukaryotic cells is basically a cell that is a little bit more complex than a bacterial cell, okay? They have a nucleus, they have membrane-bound organelles, and they do some metabolic functions. So we saw all of a sudden the race of this early eukaryotic cell, and where did it come from? Okay, that's point number 4. And therefore you have to look at this image. What is very interesting is that if you look at the plant cells of today that we just analyzed, and the animal cell that we have today, and you look to the bacterial cell, then there's 2 components that are very interesting, which is the mitochondria and the chloroplast. Here, the mitochondria and the chloroplast. Now, basically, what is the whole point of this endosymbiotic theory is that we had a simple bacterial cell, an ancestral prokaryote cell, okay? Conditions were improving, so more species of bacteria would come into existence, okay? We had oxygen, so we had aerobic bacteria, that were bacteria that used oxygen to their benefit. But then after more time, those bacteria, they thought that it would be a good idea to work together. I mean, they were not conscious about this, but a certain bacteria was engulfed by a bigger bacteria, and they created a kind of symbiosis. You know what a symbiosis means? What it really is? So, for instance, if Cecia has a strength of 1 and Gustavo has a strength of 1, if you both work together, you can have a strength of 3. That's a symbiosis. It means that doing things together has, is more productive than doing things on your own, and more than double, in fact. That's a symbiosis. And that's what happened, you know. Different kinds of bacteria were into existence. One bacteria engulfed another bacteria, they ate it, or another thing that is interesting, we don't know exactly how this process happened. It could be engulfed. But on the other hand, what I think is that one bacteria might have infected another bacteria, because this is thing that we see all the time. It's still happening. We are getting infected by bacteria. Our cells are infected by bacteria every time we get ill. Nowadays, a lot of people get infected by viruses. It's a mechanism that's been there forever. People get infected by coronavirus. People get infected with bacteria, get infected with other microorganisms. So it's something that life has been doing since the beginning, is infecting each other. So possibly one bacteria infected the other one, and it was not negative, it was positive. You know, it's possible that, that they started to work together, and that this symbiosis, the fusion of the two, that this gave the rise to becoming a more complex cell, basically the early eukaryotic cell. And then in another stage, it could be that another bacteria infected the cell, and it was a photosynthetic bacteria. And then you got a plant cell, okay? It's way more complex than that, but what you have to remember is basically that the endosymbiotic theory is about different kind of prokaryotic cells that fuse together due to eating each other, infecting each other, or just engulfing each other and working together, but that it was a beneficial outcome. Because not everything that happens in life is bad, you know? Everything happens for a certain reason, you can say, and nature does things for a certain reason. With the energy we have around us, we always go to more complex and to more interesting structures. So this is basically the theory that explains how in fact very simple bacterial life became complex cells, and then further on plants and multicellular organisms. Okay, so that's basically it. What we do know is what we also know, what is some evidence about it, is that the mitochondria, they have been analyzed very carefully, and do you know what they found in the mitochondria? And they didn't have an explanation for it, but now it's an evidence that indeed this endosymbiotic theory is the most likely thing that has happened. What do you think they found inside mitochondria that seems not to have any function anymore today?
A nucleus?
Yeah, not really the nucleus, but what's inside the nucleus. So what is that?
DNA.
Yeah, exactly. They found DNA in mitochondria, and it seems we don't know what the function is of that DNA. It's very simple, short DNA. It looks exactly the same as bacterial DNA. So it looks that the mitochondria is like the evolution of a bacteria that was engulfed by a cell. Same thing for chloroplasts. They analyzed this chloroplast into detail, and they found DNA in the chloroplast. They found DNA in the mitochondria, and it doesn't make any sense. It only makes sense if this is still characteristics of millions of years ago that are still there, but that don't really have a clear function anymore today. Okay, or maybe they have, and we still don't know exactly. But they're investigating this. They're investigating if this DNA still has a function or not, but it's a clear evidence that something happened millions of years ago and that certain bacteria started to work together in order to become complex life. So it's way more than a theory. Also, if you look to the mitochondria, if you look to the chloroplast, if you look to these entities, to these organelles on a very specific level, and you look at them, how they work, they have very, very remarkable similarities with bacteria that we still have today. And you know that this cannot be a coincidence. This means something. This is an indication of something. And this is all proof that this endosymbiotic theory is the most plausible thing we have today to explain how complex life came into existence. Okay, and I completely agree. I think the evidence is overwhelming. I think we can be almost absolutely sure about this endosymbiotic theory to be true. The only thing is that we don't know every little step about this theory yet. So still things have to be discovered. So we don't know exactly how the cell went into the other cell or which kind of bacteria fused together. It's all up for debate. You know, there's still things that are unknown, but everything seems to indicate that this is what happens. And if you want to be a good scientist, you always try to be as actualized as possible, and you can follow this in scientific newspapers to see if there is any evolution in this. But for you, it's important to know, you complex person, because we all are, where do you come from, and how did it all happen? You know, if you start thinking about it, the time that we all had to evolve into what we are, it all makes sense, you know. So any questions? If not, that's it for today, and we see each other tomorrow, and then we go into more detail about this theory. Or somebody, did you like the conference, by the way?
I have a question.
Go ahead, Adrian.
Could it be possible?
Yes.
Yes, I love her.
Sorry.
Could it be possible, like, to make a machine, or I don't know, to?
What kind of machine, Adrian?
Well, like, I don't know, like it combines gases in fusion. I don't know.
Yes, I think it would, but it's not something. Look, this Miller-Uller experiment where they had like atoms and they automatically formed biomolecules, it was something on a very small scale. But if you want to make a machine that makes life, it's not that simple, because life took millions and millions of years. You cannot expect this to happen in experimental conditions, you know, in the short time we have. It is possible, but it's not that simple to make a machine to make cells, because it's too small, to be honest. We don't have the technology. How do you want to do with metal and plastic make life? It doesn't work like that. Life can only be made by life, and that's what we will see next lesson. That's part of the cell theory. So we are still far from that, from making life with machines. It's not that simple, because machines are dead and life is alive. And maybe we get there one day, but we can still not do it perfectly. All life we make in the laboratories today is just the machines. They make the conditions to make it possible, but it's life itself that is producing life, not machines. And sorry, yeah, so I'm glad you liked our guest speaker. So I chose her for a reason. And it's not only because I'm married to this girl, but also because she is inspirational, you know. She was one of you, and she just decided she wanted to do something for the world. She wanted to see the world, and all of you can do the same. So I think it's really nice to put a Mexican in front of you that did different things, you know, and didn't follow like the standard procedure, let's say. And you all can do that. Dream big and do big. So that's why I asked her, please tell them this. In fact, it's the first time I put her, because now of the contingencia, it's difficult to find persons to do that. But yeah, I'm glad you liked her. And yeah, if you want to do it again, we can set up another meeting one day. Normally there is still one other guest speaker for next semester. No, next partial, possibly. So I will keep you up to date. And thanks for being here. And if there's no more questions, then we see each other tomorrow, because otherwise I'm passing my time, I see. So we're all
Thank you,
good?
teacher.
Okay.
Thank you, teacher.
Thank you.
Thank you.
Thank you,
Thank
Mr.
you.
Steve.